All right. Well, thanks for that great introduction. I want to thank our hosts for organizing this wonderful meeting. This has been such a great experience for me to interact with clinicians and patients and other scientists. And so thank you very much for this. It's a great honor to be here. And so I'll talk about my lab, and I'm on the clock, so I'm going to go fast. But my lab, we study the cerebral circulation, and we're really interested in the basic mechanisms by which the cerebral circulation is regulated. And so the brain is a unique organ. It's unique in many ways, but one of the ways hydraulically it's unique is because it's within a skull. It's the only organ in the body that's actually within an inelastic compartment. So like if the brain needs more blood, you simply can't pump more blood into the brain. That would be bad. The brain would swell up. It would press against the skull. The top of your head would pop off, and you'd be in a lot of trouble. So there are mechanisms that auto-regulate blood flow to the brain. And it's one of the most constant physiological variables in the body. The global amount of blood entering the brain and leaving the brain doesn't change very much over time. But because of this, when brain regions become metabolically active, blood's got to be delivered in real time, and like an on-demand system. So it's an on-demand system, and this is called neurovascular coupling. And so this is basically the neuron signal the blood vessels to dilate and to deliver more blood where it's needed most. And so my lab has been studying these basic physiological processes that are not completely understood. We've been studying these from the molecular and cellular viewpoint for maybe about 20 years. And this is important. And so, what happened? I'm missing a slide here. Oh, well. All right, so neurovascular coupling, this is the matching of blood flow to the metabolic demands of the brain, and this is what we're going to talk about today. And so one of the ways that we can study this, we can demonstrate neurovascular coupling, is by using this mouse model. And so it turns out in mice, it's really fortunate in that the barrel cortex of the brain, which is on the surface of the brain in rodents, is innervated by the whiskers. So if you go in there and you tickle the whiskers, you can induce functional hyperemia right in the top of the brain. And because of this, you don't even have to make a cranial window. You can just thin the skull down a little bit, and then you can use a technique called laser Doppler fluorometry to measure changes in blood flow. And so then you can see that in some of these data. So you can see where the whisker stimulation is. You can see this increase in neuronal activity in the barrel cortex, and then you can see the increase in local cerebral blood flow measured using laser Doppler fluorometry. So this is a basic response that we're going to talk about today. This was discovered over 100 years ago by Sherrington and Roy. There's a picture of those guys taking a smoke break after doing some experiments. And so the reason why we're interested in this from a translational standpoint is because dementia is a vascular disease. So we know what dementia is. It's this progressive loss of cognitive function. It's age-related, so because people are living longer and longer, there's a lot more dementia in the world. We proved that in our last election in the United States. Alzheimer's disease and cerebral small vessel disease are the leading causes of dementia in the world. And so cerebral small vessel disease, we know that that's a vascular disease. We've talked about that some today, so I don't have to belabor the point. But at least 50% of Alzheimer's disease patients show vascular injury. So at least half of all of Alzheimer's disease, dementia associated with Alzheimer's is called mixed dementia. So about 30% is mixed dementia, about 20% is pure vascular. So half of all dementias are vascular in origin. And so we know that cerebral blood flow regulation, the processes that I talked about, autoregulation and neurovascular coupling, they become deranged during cerebral small vessel disease and Alzheimer's disease. And so the big question that my lab has been pondering is, can we restore cerebral vascular control to halt or reverse cognitive decline? So I was sitting in my office pondering these questions, and then this guy Doug Gould comes wandering into my office and kind of out of the blue off the street, and asked me if I knew anything about blood vessels. And I said, well, you found the right guy. And so we formed a partnership, on basically that one day, and we wrote a couple of grants that got funded, and we've been studying this for the past 5-6 years, something like that. And so we've been studying this allelic series of COL4A1 mutant mice that Doug talked about earlier in the context of cerebral vascular control. And so this work was largely conducted by Dr. Evan Yamasaki, who was a fabulous PhD student in the lab. He did the bulk of the work that I'm going to talk about today. All of the animals that we studied were heterozygous for these COL4A1 mutants. I'm going to talk about 2 mutants. Mostly I'm going to focus on the one that's G394V mutant, but I'm also going to mention our work with the 1344D mutant. These animals were all either 3 months or 12 months of age. And so we found that this was an age-dependent phenomena, at least in the mice. So this is important. It's important to note that 3-month-old mice they're young adults, so they're like graduate students. So they're like 20-year-old people. 12-month-old mice they're not old, they're middle-aged. So they're like people in their 40s and 50s. They're like associate professors. So, that's the middle age of mice. We studied males and females in equal number. We tested for sex differences. We didn't see any sex differences. So I worked on this project with Doug, of course, and with Miriam Chamel, who was at the time we started this project was at UCSF. And Miriam's a great neuroimager and did some beautiful work with MRI that I'll show you a little bit of. So anyway, that's the paradigm. And so we started off looking at these G1344D mice that Doug mentioned. And, and this has the mutation, down near the NC1 domain. And these mice had severe age-dependent brain pathology in our hands. So we did Prussian blue staining. You can see, in the top panels, the pink stuff. So the blue staining is Prussian blue. That just shows you that a hemorrhage occurred there at some point in the animal's life. We saw a lot of pathology, and then Miriam did the neuroimaging, and we saw a lot of brain lesions. You can see these are MRI studies, and you can see the 12-month-old mutant mice had quite a few lesions. The wild types didn't, and this was all age dependent. We didn't see any of this in the 3-month-old animals. We did similar studies in the other mutant, the G394V mice, and what we found in these mice was interesting. It was very different. We really struggled to find any pathology in these animals. So in the young animals, we didn't see anything. And the old animals, in a few animals, when we did the Prussian blue staining, we saw some very low levels of hemorrhage, but not even really enough to quantify. We saw a few animals that showed some bleeds. And the MRIs were unremarkable. I mean, we didn't see any pathology in the MRIs. So we thought, we were a little bit disappointed. We were like, do we continue to study these animals? And we decided to do one more experiment. We did some behavioral testing on these animals. And so we did this experiment called Y maze. And so this is just a standard test where you put a mouse in this Y maze thing. And the wild-type mice, they will explore each arm equally. Mice are really curious, so they just want to check out each arm equally. Whereas if a mouse has some form of cognitive impairment, they'll ignore one of the arms, perhaps, because it's kind of like when you walk into the room and you walked in there to get something and you forgot what you got, what you went in there for, so you go back out again. So it's kind of that same process. So what we found was the 12-month-old G394V mice had some form of you can see it here. Let me see. I can't see the pointer. There it is. So you can see here, these mice, they spent less time. They basically kind of alternated between 2 arms instead of spending time in all 3 arms. But they walked the same amount of distance, so that there was no problems with how they were moving around. We did a similar assay. It's called a novel arm assay, where you block off one of the arms, and the mice tend to want to go to and then the next day you open it up, and the mice tend to want to explore the novel arm more because they haven't seen it before, so it's new to them, so they usually spend more time in the novel arm if they're normal, but if they have some form of cognitive impairment, they'll avoid the novel arm. And we saw that, again, there were deficits in these animals. So these animals don't have any overt pathology. We can't see anything on an MRI scan, yet they're showing signs of dementia. So I thought this was pretty interesting, and this may this may inform some silent cases of dementia that we really don't know where they're coming from. We think that these defects are resulted from impaired neurovascular coupling, so we set about to study that. And let's see, I'm having trouble here. So I need to tell you a little bit more about neurovascular coupling. And so neurovascular coupling is regulated in part by the capillaries in the brain. So the capillaries in the brain, so you can see from this just this representation up here, this is a 1 square millimeter just of a section of a random brain. And you can see that, the arteries are in red, the veins are in blue. All that gray stuff is capillaries. So the capillaries it's just incredibly dense. There's probably 1 capillary endothelial cell for every neuron in the brain. So the capillaries, and they're right next to the neurons, so they're well positioned in order to sense what's going on within the neurons. So the capillaries, in a nutshell, you can see in the diagram, the lower diagram here, in a nutshell, what happens is that the neurons drop stuff onto the capillaries that's sensed by particular molecules in the capillary endothelial cells, and they orchestrate the dilation of the upstream parenchymal arterioles in order to locally increase blood flow to meet the metabolic demand. So there's 3 different pathways that have been studied so far. There's inward rectifier, there's TRPA1 channels, and then there's prostacyclin receptors. We're going to focus on the inward rectifier for the purposes of this talk today. So this is the heart of this capillary to arterial dilation process, so we need to talk a little biophysics. So, and these are really unique channels. The inward rectifier has 2 unique properties that you need to be aware of to understand how they regulate neurovascular coupling. Number 1, these channels are activated by small increases in extracellular potassium. So we're talking like if extracellular potassium goes up from a normal value of, say, 3 millimolar, it goes up to 10 millimolar, that opens the channel. It opens the channel because it pushes out polyamine and magnesium block. So the potassium ions enter the pore, they remove this block, and then potassium ions can flow out. This has a whopping hyperpolarizing effect on cells that express these channels. So they're activated by extracellular potassium, and then they're also activated by membrane hyperpolarization. So hyperpolarization of the plasma membrane also pulls out the magnesium block and the polyamine block, and that activates the channel. So that allows them to propagate signals throughout cells that are electrically coupled. So for example, if this cell, if we dump some potassium on this cell and activate the inward rectifier, hyperpolarizing the membrane, it's going to hyperpolarize its neighbor, and the signal will propagate if these cells are tightly coupled and not leaky. So basically, that's how this process happens. So we thought the inward rectifier was really important, so we decided to use patch clamp electrophysiology to measure these currents. And what we found was that, all this work is done in native cells. So we isolate chunks of the brain, and then we mash it up, and we're able to pull out brain capillary endothelial cells, and we study them within a few hours of when we isolate them. So none of this work is done in cultured cells. We found that there were deficits in the inward rectifier channel in our 12-month-old animals. At 3-month-old, currents were okay. At 12-month-old, we can see that the currents are deficits. So they're very low in both brain capillary endothelial cells and arterial endothelial cells. So now we know that this collagen mutation results in an age-dependent loss of the inward rectifier potassium channel current, which is probably one of the basic disease mechanisms. We also wanted to look at the physiology of this, so we used ex vivo pressure myography. This is a neat technique where we take blood vessels, we take these tiny parenchymal arterioles out of the brain, we put them in a pressure myography chamber, and if you want to have a look, they kind of look like this. You see the parenchymal arteriole, the capillary bed's down here, and then we have this weird zone called the arterial to capillary transition zone, or ACT zone. And then we can stimulate the capillaries with stuff from a picospritz or pipette. So we picospritz stuff onto the capillary, and then we see what happens to the arteriole. So these arterioles are 25 microns in diameter, so these are really hard preps to do. So there's only a few people in the world right now who are doing these. So this is what a typical experiment looks like. So this is just a movie of, you can see our pipette here. We're going to pump on some potassium. So you can see the potassium's applied to the capillary, and then it causes dilation of the upstream arteriole. And then after this signal washes out, the arteriole constricts back to its normal diameter. So this is a demonstration of capillary to arterial dilation. And we've done all the proper controls to show that this is truly a conducted vasodilator response, and you can look up our paper if you want to see those controls. So when we did this experiment in our mutant mice, what we found was in the young mice, obviously everything was fine. In the 12-month-old mice, this response was gone. I mean, just flat out gone, did not exist anymore. So you can see that these are traces of some of these experiments here. We see in our wild-type mice, we get a nice dilation every time we pump on potassium. But when we do it in the mutant, we saw no dilation whatsoever. And at the end of the experiment, we dilated the arteriole by putting on a nitric oxide donor just to show that the arteries can dilate, but they don't in response to the potassium. So this was really interesting. So then we looked at the functional hyperemia response. So we looked at the blood flow response in the brain, again, using our laser Doppler approach. And so again, this response was significantly blunted in our 12-month-old mutants. So you can see this is this orange trace here versus the black. We stimulate the whiskers, in this case, for 1 second. And we can see that the blood flow response is significantly blunted. It's almost gone. So these animals have an impairment in functional hyperemia, at least in the barrel cortex. We wanted to know why. Okay, why is the inward rectifier gone in these animals? And so we started to think about this compound called PIP2. So PIP2 is a minor membrane phospholipid. It's required for the activity of the inward rectifier channel. When we first looked at mRNA levels, there was no difference in mRNA. So then we started looking at PIP2. And so when we added PIP2 exogenously to the patch clamp pipette, we just supplied it, we overdosed it with PIP2, we saw that the currents were fully restored. So that's telling us that somehow this collagen mutation is causing a deficit in PIP2 in these endothelial cells. So why? We want to know why, so we went upstream a little bit. We first thought it might be this enzyme called, at first we thought it might be decreases in levels of ATP. So, ATP, the current energy currency of the cell. Synthesis of PIP2 requires lots of ATP. So we measured ATP levels. They were not different, so it's not an ATP deficit. This enzyme phospholipase C, or PLC, that cleaves PIP2 into a molecule called DAG and another molecule called IP3. This is a ubiquitous enzyme. It's in every cell. We thought, well, maybe there's elevated levels of PLC, but that wasn't it, because we blocked it with this drug, and that had no effect on the current deficit. So then we thought about this enzyme called PI3 kinase. And so PI3 kinase is an enzyme, another ubiquitous enzyme that basically converts PIP2 to PIP3. It adds another phosphate onto it. And PIP3 doesn't have the same activity as PIP2, does not stimulate the inward rectifier. So we used this drug. There's really good drugs for PI3 kinase, and we picked this one. This is a pan-specific PI3 kinase inhibitor. So we blocked PI3 kinase and we restored the currents. So this is telling us that somehow these collagen mutations are causing an increase in the activity of PIP2, or an increased activity of PI3 kinase, which is causing PIP2 depletion. We took it back to the level of the pressure myograph, and we found that in the same vessel, we initially, this is the same arterial. So initially, before we treated it with anything, again, this response to potassium was completely eliminated. But when we treated for about 20 minutes, we could fully restore this response. So this is telling us again that this PI3 kinase is probably what's causing the PIP2 depletion, and that's what's causing the loss of capillary to arterial dilation. What's driving this? Well, we think it's probably TGF-beta. Doug talked to you about TGF-beta being elevated in some of these collagen mutant mice. We know that TGF-beta can activate PI3 kinase through this TRAF6 ubiquitin ligase pathway. So we tried an experiment where we blocked TGF-beta receptor, and again, this restored the capillary to arterial vasodilatory response. So now we're getting somewhere. So now we think that TGF-beta is increasing the activity of PI3 kinase, depleting PIP2, and then we lose the activity of the inward rectifier, and that's what's causing the neurovascular coupling defect. We wanted to test this out in vivo, so we did the following experiment where we injected mice basically daily with our PI3 kinase inhibitor. So on day 0, we did a Y-maze assay on these mice. And then we started injecting them every day with this drug for 28 days. And then at the end of the experiment, we repeated the Y maze and we did a bunch of ex vivo experiments. And what we found was that we completely restored everything. So, I mean, this basically fixed the problem. This fixed the current deficits, so you can see this. These are patch clamp experiments. So in our vehicle-treated animals, we had a massive current deficit. This comes back if we chronically block PI3 kinase. In terms of capillary to arterial dilation, again, it's gone in our vehicle-treated animals. It completely comes back in our animals that are treated with the PI3 kinase inhibitor. If we take it in vivo, these are the blood flow measurements using laser Doppler fluorimetry. Again, we get a blunted signal in our vehicle-treated mutants, and then it's not quite fully restored, but it's about 80% restored in our animals treated with the inhibitor. And then when we go to this test to see if they have cognitive impairment, we see that there's actually a pretty good improvement in their cognitive impairment scores. This is the important group to look at here. We see that there's actually an increase in this alternation index in these animals when they're treated with the PI3 kinase inhibitor, indicating that they have improved cognitive function. So the fly in the ointment here is that there's adverse effects. There's pretty severe adverse effects of these PI3 kinase inhibitors. One of them that's been described in human patients is fatigue, and we saw that again in our mice. We saw that the mice didn't want to move as much. So this shows you the distance moved, and this is both for our wild type and our mutant mice were equally affected. They moved quite a bit less than the vehicle-treated animals. So just to sum it up, what we think is going on is that somehow in this particular mutant, we're getting increased TGF-beta activity that's driving PI3 kinase, depleting PIP2, and that's silencing the inward rectifier. So that's leading to reduced neurovascular coupling and functional hyperemia, which leads to these impaired behavioral outcomes indicative of cognitive impairment. If we can block this pathway in our case with a PI3 kinase inhibitor, we can restore all of this. So just a few final thoughts on the therapeutic potential of PI3 kinase inhibitors. So, we showed that PIP2 depletion contributes to cognitive impairment in our collagen mutant mice, but this has also been shown in the CADASIL cerebral small vessel disease model, as well as in a model of familial Alzheimer's disease, the 5XFAD model. So it looks like PIP2 depletion might be kind of a universal marker for several types of cognitive impairment. There are inhibitors. There's about 5 inhibitors that have been approved for use in the United States. There's many more that are kind of, in clinical trials. And I guess the downside is that, they have adverse effects like fatigue and some GI issues. But, these drugs may hold promise for treating dementia associated with cerebral vascular dysfunction. So I think this is pretty interesting. And then just the last slide to thank everybody. Like I said, most of this work was done by Evan with help from Pradish, and it was initiated by Harry Pritchard and Paulo Perez. Paulo gets really excited at baseball games. These are my dogs. They don't care about any of this, and got to thank Doug and Miriam for all their help and the funding agencies. So thank you very much.
Okay, thank you. I was just saying it's really interesting because it's connected with so many other diseases of the pathway. So there are so many insights about that. 2 questions.
Thank you so much, Scott. It's so wonderful to see the story. So 2 questions. One is length of treatment and at what stage of development do you get these clinical effects in your mice? And the second one is, we know we cannot go at these pathways like TGF-beta, PI3K systemically, localizing the drug treatment to the cell type, and whether or not also go after TGF-beta trap. They're now clinically ongoing for pulmonary hypertension, very similar mechanism. What are your thoughts about this?
Well, in terms of your first question, the length of treatment, we started at 11 months, and we saw that they had cognitive impairment already by 11 months. So, and then we treat it for a month. And we haven't tried for shorter periods of time. We haven't played around with a time frame on that. It's just really, these are really labor-intensive experiments. It's hard to get postdocs to come in and do injections every day. So, and then in terms of in terms of improved therapeutic development, that's one of the reasons why I was excited to give this presentation. Hopes that people who know, people involved with this will, maybe get excited about using this approach. I mean, it has to be refined and it has to be improved. I mean, we can't do what we did to the mice to people. I don't think that would be a good idea. But improved ways of targeting these things. The TGF-beta, I mean, that's another way. The TGF-beta receptors have not been well tolerated in clinical trials, so that, that's out. The PI3 kinase inhibitors are sort of a last resort for cancer, so I don't know that they're really viable, but if we could find ways, I know that there are more isoforms. One of the things we want to do in the lab is to identify the particular PI3 kinase isoform responsible for this, to see if we can, maybe use a targeted inhibitor. And then we're also targeting this pathway. We made a PI5 phosphatase kinase knockout mouse, so we want to deplete PIP2 just genetically to see if it recapitulates a lot of these things. So that's some of the directions that we're going in. I mean, but we would love to involve somebody who wants to do more development on this.
Very beautiful talk. Quite amazing, the effect on memory. So the why maze experiment, I guess you are mainly testing short-term memory.
Yeah, spatial working memory.
Exactly. Have you also thought about or performed the experiment for other behavioral paradigm where you test long-term memory or other form of memories? And if you have any thoughts or results?
We would love to do that. We want to do that. That's one of the reasons I just moved institutions from University of Nevada, Reno to University of Rochester, because they have a behavioral core that we can take advantage of. I mean, we're electrophysiologists, and we said, okay, well, we have to do behavior, so we're trying to teach ourselves how to do this. And so, but yeah, I mean, your comment's well taken. I mean, there's a lot more work that should be done,
Because it's quite amazing that, I mean, short-term memory doesn't really require anything but the proteins they already have. It's mainly post-translational modifications. Right. But you have a beautiful effect, a really strong effect. Right. And I don't know, it's quite surprising. Maybe not even affect synaptic plasticity, maybe just something else. It's quite amazing.
Yeah, I mean, we're pinning it all on improved neurovascular coupling, and improved vascular function. But yeah, it could be something else. I mean, we simply don't know. This is new for us. I was very surprised. I was really skeptical that experiment would work. And we repeated it many times. We did it blinded. We had different. We gave the data. We gave the videos to different people to analyze. And we did everything we could to ensure rigor. And and then sooner or later you have to believe it.
Yeah, absolutely, but from a physiological point of view, you may have a very nice model to really discover something else, mainly about neurons, really. Yeah. It's quite impressive, really. Beautiful.
Thank you.